afternoon, and maybe good morning for some of the participants. I'm very happy to see that the audience is really diverse, as Philip said before. I think that we have more than 10 different breeding companies represented, and it was one of the things we wanted to achieve with this second event, to dig or not to dig, on discussing about the options available or options to take while considering the place or the improvement of root rates in agriculture. So I will quickly set the scene from a scientific point of view. So I'm not going to make a long introduction of roots. I think that if you are there, you know what they are about, and the topic that Jonathan will address will actually make it really broad. So when we started and we designed the SolACE project, we had a very big ambition in terms of phenotyping strategy. It was in 2015, so time is going on. So we had in the project, we have been working on 3 species: potato, durum, and red wheat. Today we are going to talk mostly on the durum and red wheat. And one of the work package in the project was dealing with genetics, which led us to work with large panels, and we chose to work with diversity panels of more than 200 lines in red wheat and in durum wheat. And these 2 panels have been kind of intensively phenotyped using state-of-the-art phenotyping platforms that are available in Europe. And they are also they were also ground in field in a small field network. We had 4 to 6 trials depending on the species. This was generating a lot of data, and we knew it. We actually wanted this data to be and we knew we would face some challenge in connecting information coming from platforms in controlled conditions on very detailed traits and younger plants with the response of these the same genotypes in field conditions. And because of that, we were all convinced that one way to go and connect these different variables was to use a model and to develop a new kind of models. So the root platforms that have been mobilized in the project that I mentioned is 4PMI in Dijon. It's a high throughput root phenotyping platform based on rhizotubes, and you will hear a bit more about later. Another high throughput root phenotyping platform was the RootFair aeroponics platform in Louvain-la-Neuve. And this was complemented with lower throughput experiments in PVC tubes, where roots could be measured in real-like substrates, plus the Radimax setup that was one of the main points in the field and discussed in a previous event. So we are unfortunately not going to talk about Radimax today, except in the discussion. So what we expected when we collect all those data and try to assemble them, so we had again 250 genotypes. That was reasonably large to estimate genetic correlation, for example. Well, we tried the crude and simple thing of correlation matrix between all these variables. And so here we have this large correlation matrix with a first block here corresponding to the variables. We had 12 different variables estimated in aeroponics. We had the 2 sets of nearly 20 variables estimated on pouches. We had at that time 6 variables estimated in the tubes in Dijon. We had another set of highly specific variables on root diameters in PVC tubes, and there was one critical summary from the field network that was an index of sensitivity to water deficit and nitrogen deficit. And so we expected that it would be difficult. I must say we hoped there would be something very easy to take out of this analysis. And well, what we see here is that when you go in area of the correlation matrix that is between variables of different experiments, we have virtually no correlation that pop up, and especially when it goes to linking platform data with field sensitivity, yeah, it was a bit depressing. So, but these are the data, and we have all worked with state-of-the-art installation and field experiments, following all the recommendations from consortiums in phenotyping. So we believe we did a good job. So these has actually defined the topics we wanted to share with you, because these results are going to be published and you will know about them. But having a discussion with the stakeholders about what we believe are hot topics in the area of bringing root phenotypes into a breeding context. One is, what about this difficulty to handle root genotypes that have tend to have a low reproducibility? Is it a matter of noise? Is it a matter of plasticity? Can we control that? And another question that came out to be really crucial is root traits are not all equal. And every of those platforms, every of the experiment that we can see in the literature use a set of root traits that come from, well, various motivations. And this brings me to the introducing the different speakers. So Jonathan Lynch from Penn State University is going to spend half an hour on making a wrap-up, I would say, of his entire career, with two underlying messages that it is possible to, there are different ways to dissect phenotypes, but it's possible to identify right traits that he is naming phens that are components of a phenotype and that can have the right properties to be amenable to breeding. And also Jonathan will make an update on root traits phenotyping options. Well, that will go through his presentation. And after that, we will have 4 flash talks from SolACE participants. One by Christophe Salon and Renaud Rinson about their approach to try to link field and platform using QTLs and QTL colocalization. That was an example in breadwheat with the 4PMI platform. Another one by myself, Gabriela Dono and Nicolas Peccioni on durum wheat, using root QTLs as ingredients for genomic selection. Then Boris Parent is going from INRAE, is going to talk about the interesting correlation between or links between tillering and yield sensitivity. And we know that tillering is probably is certainly a key point in terms of root architecture on the later stage of growth in wheat. And then Pierre Martre is going to illustrate the attempts at what we are currently doing still in the project to connect virtual plant model and crop model. And we will end up with a long time for discussion. So I will directly leave the floor to, so I stop sharing and leave the floor to Jonathan. Jonathan, I thank you again for agreeing to spend a lot of your time with us, and the floor is yours. Thanks.
Thank you very much, Xavier. Can everyone hear me okay?
Yes.
Yes, Xavier? Okay. Thank you very much for those comments, Philippe and Xavier. And thank you all for joining today. I'm very honored to be able to speak with you. Let me begin by saying that I believe you young people are facing tremendous civilizational challenges, obviously, supporting 10 billion people in a degraded environment. And I believe understanding root phenotypes is an important technology to address those challenges. So I respect the task you have undertaken, and I'm very honored to be able to speak with you. So as Xavier mentioned, this is a very brief overview of some of the themes of your meeting today. Xavier told me to address these points, these 5 points. Now, each of these points is important and complicated and worthy of much discussion. I will just give some brief examples from our own work, hoping to use that as a starting point for some discussions, perhaps. I will be here with you for the next 2 hours to join in discussions. But if any of you, especially young scientists, have any interest in further discussions or clarifications, please reach out to me, and I'd be very honored to talk with you further about these issues. So the first topic I was assigned, root phenotypes are keys to crop function and response to low nitrogen and water. Now, again, I will not attempt to summarize a rather large literature on this topic. I will just give you an example of our own work with one of the species we have worked with, maize. So here we see some phenes. Phene is an element of a phenotype that we have worked with in terms of yield of maize. Now, this is yield in the field, mainly in North America and Eastern and Southern Africa. And you can see yield under drought, yield under nitrogen stress, yield under phosphorus stress. And we have 7 different phenes, anatomical and architectural phenes in the left column. And this is all published, so I'm not going to dwell on this, but I just want to point out that we have seen some very promising results. So I highlight in yellow here, these are yield increases. So we've seen up to 8-fold increase in yield under drought due to a root phenotype, root cortical-parenchyma. But these numbers that are highlighted in yellow, each of them is over a double increase in yield under stress. So I think these are very promising avenues to breed more stress-resilient crops. Now, this is a different species. This is common bean, the most important food legume on Earth. And we worked for many years breeding ourselves and working with bean breeders in Africa and Latin America to develop bean lines with better tolerance to low phosphorus availability, which is a key constraint to bean production in developing countries in the tropics. And so on the left, you can see a typical bean phenotype, a standard phenotype, the phenotype that is commonly found when we began our work back in the 1980s. And on the right, you can see after many years of work, we discovered various phenes states that were contributing to topsoil foraging, which is important for phosphorus capture because most of the phosphorus availability is in the topsoil. So you can see it's not just one phene state, but multiple phenes we're interested in here. Many of them architectural, but also anatomical. You see at the bottom root hairs. And employing phenotypic selection, direct phenotypic selection, because bean breeders in the tropics generally do not have money or resources for genotypic selection. But these are all visible traits that can be easily seen, visible phenotypes. So selecting for these root phenotypes, we have ourselves and in collaboration with CIAT and African breeding programs, released new bean lines with better root phenotypes for topsoil foraging that have between 100 and 150% better yield in regional trials in Africa and Latin America. And for example, 2 years ago, we've released 6 new bean lines in Mozambique with substantially better yield in low input conditions than the best available elite lines. So this demonstrates the value of root phenotypes for breeding. We also have done this in soybean in collaboration with my colleague Xiaolong Yan, who's tragically passed away from cancer some years ago. But taking the same ideas from common bean, applying them to soybean, which is a genetically related taxa. You can see on the left, I'm not sure if you can see my pointer here, but on the left, the far left, you see a standard soybean root phenotype after several thousand years of soybean cultivation in China. On the right, you can see this topsoil foraging idiotype that we were selecting for. And then the image on the right, you can see the result in the field in South China. South China has low phosphorus soils. And compared to the best available varieties, these phosphorus efficient lines developed by Xiaolong's team had much better yield. 15 phosphorus efficient soybean lines were released by Xiaolong's team in China. 3 of those actually were released in Mozambique. So our work with legumes demonstrates the value of root phenotypes for breeding for adaphic stress. The second point, how do we assess large numbers of genotypes? I'm just going to give you this one slide. Of course, the throughput of phenotyping is totally dependent on how you are phenotyping, what is your situation. But I can say that over the past 15 years, our own breeding and genetics work, we normally achieve between 100 and 200 genotypes per day in the field for multiple root phenotypes using a crew of 3 to 10 laborers, and these are generally unskilled laborers. And as I'll talk about in a moment, this rate is being accelerated. We have similar throughput for seedlings in controlled environments, but with less labor. And in our hands, this throughput is sufficient for breeding and for genetic analysis. The third point, root phenotyping in controlled environments, and I believe this project has considerable expertise in this, and you'll be hearing some other presentations today about sophisticated controlled environments. Our work has always focused on very simple technologies because we're interested in helping third world breeders who don't have access to a lot of money and equipment or even electricity, for example. So we do very simple things. So I'm going to tell you a story about a very simple study. Now, this is with common bean, and we were interested to know if the architectural phenotype that we see in seedlings in growth pouches, a very simple, blue paper, blue germination paper in a plastic bag, accessible to anybody on Earth. And we evaluated a diversity panel of 577 bean genotypes representing the entire diversity of cultivated common bean in over 50 environments, representing 5 years of trials, 3 continents. And you can see on this map a wide range of locations in which we had either non-stress, drought, heat, or low fertility. And we were interested to see if our simple seedling phenotypes had anything to do with yield in these various environments. This is work of Chris Strock, and this was published. And so by screening all these genotypes in the seedling stage, we were able to cluster them into 5 groups based upon, for example, basal root number, taproot length, other things. And Chris identified clusters 3 and 4 as being sort of intermediate in terms of having both topsoil exploration, which is important for phosphorus capture, and subsoil exploration, which is important for nitrogen and water capture. And if we look at the yield of these plants over these many trials I mentioned, we can see that clusters 3 and 4 had the best yield under non-stress conditions. Cluster 4 had the best yield under drought. And clusters 3 and 4 had the best yield under low fertility. And I think there's good reasons for this. We believe that this sort of dimorphic root phenotype that can do both topsoil and subsoil foraging efficiently is optimal for this species. I won't go into that today. I know that common bean is not part of your project, and I don't think it's very important in Europe. But I'm showing you this as an example of a simple seedling screen that actually was related to field performance across a wide range of environments. So the fourth topic, root phenotyping in the field, and I think this is where most root phenotyping should happen, and it's where most breeders are going to be conducting root phenotyping. I was going to mention 4 different things here in this topic. The first is shovelomics, which I think Philippe mentioned. And this is basically taking a shovel and digging up your root crown, just the crown of the root, washing it with a hose and taking a photograph. Or just visually observing it. And a lot of important phenotypes can be observed at this scale. This normally takes us about 2 minutes per plot with unskilled labor. We normally do about 25,000 plots per year with a variety of species, as you can see here. Alex Bush, working with us, developed a nice software tool called DIRT, Digital Imaging of Root Traits, so that instead of just visually inspecting these plants, which is where we began this work, or taking a photograph for later image analysis, in this case, you could take a photograph with any cell phone or camera, and then later upload it to Alex's website, and this software will return back to the user free of charge various metrics related to that image. Let me give you an example of how this has worked. This is an example from our maize work. So these are maize crowns excavated in the field. I believe this was South Africa. And you can see in this case, we're looking at the growth angle of crown roots. So you can see it varies amongst maize lines from very shallow to very deep. In fact, when we phenotype 5,000 genotypes as part of the maize nested association mapping panel, see wide variation in angle from basically almost horizontal to almost vertical. And this is something we have always observed with root phenotypes. We see huge natural phenotypic variation for every root phenotype we've ever examined. So using Shovelomics in the field, we evaluated 400 genotypes of a maize diversity panel over 4 years in South Africa. And this particular phene I'll be talking about here is root angle. And with that information, these lines are genotyped, of course. We can conduct GWAS. We see in this case an interesting candidate locus on chromosome 1. This is work of Hannah Schneider, who's now at Wageningen. We have an expression atlas for maize that tells us that this candidate is expressed in roots. So this was interesting. And a mutant line was available in this candidate gene, CYPK. And you can see, even with your eyes, that the mutant has a different root angle than the wild type. Under this, you can see that the bars are under low nitrogen and under drought condition. The root angle of the wild type was shallower than the mutant. And so we think that under nitrogen stress and water stress, we want roots to be steeper, so the steeper angle was beneficial. This mutation was also associated with better nitrogen capture and growth under low nitrogen. So we first examined this in our root simulation platform, OpenSimRoot. So OpenSimRoot tells us that this change in angle of these roots should result in better plant nitrogen uptake and better plant biomass. On that basis, we did field studies under low nitrogen, and using N15 injected at depth, we see that in the field under low nitrogen, the mutant actually had better growth and nitrogen uptake. So this is confirming the utility of this gene or phenotype for breeding. Now we're trying to accelerate this process. I think for developing countries, Shovelomics works fine. In the rich nations, such as US and Europe, the breeding companies are uncomfortable with this kind of manual method. They'd like to see something that was more automated and objective. And so we've been developing a robot platform that is drawn by a tractor. And this is the work of Xiaomeng Shi here at Penn State. So the idea is a tractor-drawn implement would automatically lift, clean, slice, and image root crowns, so that no human intervention is needed. We've had 1 year of evaluation of our prototype. Here it is being drawn behind a tractor at our field site in Pennsylvania. Here it is in the field processing maize root crowns in the field. Once these crowns are processed, they're dropped into a cassette that you can see on the right here. That cassette is then brought into an imaging chamber that has a series of cameras and has illumination. The root crown is rotated, and basically a 3-dimensional image. And this is once again work of Alex Bush at University of Georgia. This is DIRT 3D. So this is a visual image. This is not X-ray or MRI. This is just light optics. This is just the cameras. that reconstructs the three-dimensional configuration of the crown, from which a number of root phenotypes are accessible, of interest to us, based on our research in terms of what's important for nitrogen, phosphorus, and water capture. I won't go into any detail here, but a lot of information comes out of this type of analysis that it should be useful for genetic analysis and breeding. I want to mention root anatomy because in our experience, these are very important phenotypes that are often neglected in breeding programs. And so to accelerate the process of phenotyping root anatomy, we developed this system called laser ablation tomography, which uses a pulsed ultraviolet laser to ablate tissue or anything, rocks. It could it's been used for a variety of different types of materials. You can see on the lower right a real-time image of a maize root excavated from the field being ablated with this laser. And then you're basically destroying the sample. However, you've captured an image of the sample in 3 dimensions. And so you get a lot of information and very quickly about the anatomical phenotype of your sample. And we've done some interesting things with this related to phenotypes of interest for breeding. I'm just going to give you one example. I mentioned at the very beginning of my talk that root cortical parenchyma is important for plant adaptation to edaphic stress. And so using image analysis platform that we have made available to the community called RootScan, and now that's being developed into RootScan 3D by colleagues at University of Nottingham. We can take images from microscopy or from laser ablation tomography and automatically extract various anatomical features. You can see here in red, this shows you the root cortical parenchyma area. So once again, this is work of Hannah Schneider, and we show with this anatomics platform the identification of a gene ZMBHLH121 associated with root cortical parenchyma. So with those same root crowns that we excavated in the field in Africa, we took a section of that root tissue, ablated it with laser ablation tomography, ran it through our image analysis platform, pulled out a number of phenotypes of interest, including root cortical parenchyma. So here we have GWAS for root cortical parenchyma in field-grown maize roots. We see here a candidate locus on chromosome 2. We know that it is expressed in the cortex, which is what we would like. We developed overexpression lines, RNA interference lines, and CRISPR lines to change this gene to see if it affected the RCA phenotype. Here we see RCA phenotype on the left under low nitrogen, water stress conditions or controlled conditions in the field. B73 is our wild type. When we overexpress this gene, we increase parenchyma formation. When we knock it down with RNAi, we significantly decrease RCA formation. A CRISPR line that interrupts this gene significantly decreases RCA formation. And a mu insertion line that disrupts this gene interferes with RCA formation. So once again, this is proof of concept that you can go from the field to genes controlling anatomical phenotypes in a fairly high throughput manner. I finally want to mention that a new technology hasn't been published yet called LEADER that is even quicker and faster than what I've been talking about. Here's Molly Hanlon, who's been leading this work. And this work employs handheld X-ray fluorescence spectroscopy, although it doesn't need that, but this makes it very nice. Here you can see Molly holding up this handheld unit to a rice leaf, and this emits an X-ray into the tissue or the sample, whatever you're sampling. And by looking at the fluorescence emission spectrum, you can quantify the level of metals, the concentration of metals in that sample. So you have all the metals in that leaf in a matter of seconds. So the idea is LEADER, being leaf element accumulation from deep roots, that in any soil on Earth, there's a different availability of metals with depth. So there are some metals that are found more in the topsoil for various reasons, and there are some metals that are found more in the subsoil for various reasons. And even if for some reason your soil is very young and has a uniform distribution of metals, the availability of those metals will change because of oxygen and water and microbial activity that vary with depth. So every soil on Earth has gradients with depth for different metals. So we can imagine here on the left a deep-rooted maize line, and on the right a shallow-rooted maize line. We hold up this X-ray fluorescence unit to the leaf in the field. So it's a non-destructive assay. And both of these lines should have the blue element, which is shallow, but only the deep line should have the red element, which is deep. So this is a kind of, and it can be done in any field by anybody. Now, if you're a breeding company, you can also instrument a field to make it even more precise by applying at a certain depth a metal tracer such as strontium that is cheap and non-toxic. And now by looking at the signal of strontium, you can determine if your plant is reaching that level of tracer in the soil. So Molly's been evaluating this technology across various soils and field sites in North America with diverse maize germplasm. And basically, she sees a very good correlation between these leaf elemental signatures and root depth. So D95 is taken from coring. That's the depth at which 95% of the roots reach in the soil. Here's our estimated D95 from LEADER, from non-destructive elemental analysis, and you can see an R-squared of 0.76, which is very good for field work. And she's able to very clearly discriminate deep-rooted lines from shallow-rooted lines using this method. So I think this is going to be an important breeding tool to quickly determine whether your lines are deep or shallow-rooted, which is important for water capture, nitrogen capture, and carbon sequestration. Finally, modeling, which is an important, complex topic, which I think is going to be very important in years ahead. I want to tell you the recent story as an example of what we've been doing. This is work of Ishan Ajmera with rice. So here we see on this image a version of OpenSimRoot parameterized for rice. And this is parameterized from actual field work, so it takes some time to do. This is a paper that's just been put online in Plant Cell and Environment. So we couple OpenSimRoot rice to a crop model for rice called ORISA that's able to simulate yields across various environments and various climates, including future climates. And here we see, compared to our reference phenotype here in the corner, IR64, Ishan has identified several root phenotypes which are able to significantly improve rice yield under low nitrogen supply. Here's the yield estimates. So IR64, which is one of the Green Revolution lines developed by INRAE back in the 1970s, is here under low nitrogen supply. And here are the projected yields of these new phenotypes over some past years of climate, including 80 years of future climate. So you can see at some point up to 80% improvement in yield under low nitrogen supply from some of these phenotypes. So this is obviously very useful for breeding and for prioritization of your research, to be able to quickly evaluate multiple phenotypes in multiple environments on the computer, rather than doing very slow and expensive field work. And I want to point out that some of these phenotypes do not exist or may not exist in nature. So we can create phenotypes in silico that we don't know if they exist yet, but they could become breeding targets. We can also create climates that don't exist yet, such as a future climate with high CO2 and warmer temperatures and so on. Now, an interesting feature of this study, which gets to the point that Xavier mentioned about dissecting a phenotype into phenes, is that using the simulation tool, using OpenSimRoot, we can determine which particular component or phene of the phenotype is driving our yield gain. And what we can see here is what happens to biomass for these different phenotypes compared to IR64. Here we see the greater biomass of these new phenotypes compared to IR64. Now, if we add up the individual contributions of each phene state, in other words, shallower roots or more lateral roots or whatever it is, we see that actually none of these are better than IR64. Most of them are worse than IR64. So if we just assume sort of additive contribution of different phene states, we will not be able to find a good phenotype. The reason these phenotypes are better than IR64 is because of synergism among the phenotypes. And as an example of that, I asked you to look at your hand. Fingers are wonderful, thumbs are wonderful, but having thumbs and fingers together is really wonderful. And that's an example of phene synergism that we see throughout biology. So by looking at these integrated phenotypes, how these individual phene states work together, we can find better phenotypes than than if we just consider one phenotype in isolation. So in summary, and this is a very brief summary, and once again, I'm in the interest of time, I'm just giving some examples from our own work. Other people have done nice work that we don't have time to discuss here. Root phenotypes are keys to crop function in response to low nitrogen and water. Yes. How do we assess large numbers of genotypes? Methods exist. Root phenotyping in controlled environments. I gave you the example of seedling phenotypes versus yield on 3 continents in bean that showed what I consider to be very good correlation. Root phenotyping in the field. I gave you the example of shovelomics, of root robot, anatomics, and leader. These are all deployed in the field and they're being used now to discover genes. And modeling, I gave you the example of phen synergism for nitrogen efficient rice, and models will become more available and more powerful in the future, and I believe will be very important tools for root biology going forward. Obviously, this is the work of many people I'm summarizing. This is the current configuration of our team. So thank you very much.
Thank you very much, Jonathan. I think from I knew it would be extremely informative, and I know it must be frustrating to summarize in 30 minutes all you wanted to say. There are at the moment 2 questions, and if I see them, they are kind of related to the type of discussion we wanted to have. So I suggest we keep those questions for the after the discussion, the presentation of the SolACE participants. Which then suggests that we move to the presentation by Christophe Salon and Renaud Rincent. So Christophe has made a video illustrating the 4PMI platform that is running in Dijon. So it's going to be presented, and I guess Amelia is going to share the video, and then Renaud will present the analysis of results from a wheat experiment in terms of QTL analysis and relation with the yield sensitivity to water and nitrogen.
But I don't know if there is a sound behind the movie or not.
No, the sound is coming, we checked.
Okay.
At INRAE, we have developed plant containers which allows us to grow plants and see 100% of their roots. Seeds are sown in the rhizotubes and separated from the substrate by a special membrane that allows everything, nutritive solution, rhizodeposits, microbes, to go through, but not the roots. This has been published in a paper, and this has been also patented in Europe in cooperation with 2 private companies. And these innovations are distributed worldwide. The trademark is Rhizo. In this movie, you will discover our platform, which comprises 6 environmental conditions where rhizotubes and pots are conveyed automatically from the greenhouse to the imaging cabins. We have 800 rhizotubes and 1 phytosanitary cabin. We have also 5 imaging cabins in visible light, near-infrared, and fluorescence. This allows us to extract forms from shoots. 1200 rhizotubes are automatically irrigated with various nutritive solutions. And our germination chambers are used at the start of the plant growth. We have also a high-resolution cabin which allows to get root images with a 7-micron resolution. For example, here you can see nice and precise images of the roots of a pea plant. And here is an example of what we get when we phenotype a grain legume with or without mineral nitrogen, and so respectively, without or with symbiosis within nodules between plants and rhizobia. And here you see various focus of the pea root up to a large image of a pink, healthy nodule. The second imaging cabin devoted to roots is a high throughput medium resolution rhizocab. It generates images with 42 micron resolution, and its speed allows taking images of 1200 rhizotubes per day in roughly 5 seconds per wavelength. Using these tools, we already work with many species to assess inter- or intra-species phenotypic diversity. And these various species have been studied within projects on various thematics such as plant-microbe interactions, drought-heat stress, biotic stress, nutrient stresses, or association between plants. Using these tools, we gain images which are segmented to extract the forms of interest. We can also use our algorithms to get other root traits, such as root angle, height and width, or root projected area. These help us to assess root system architecture dynamics non-destructively. As we produce thousands of plants, we had to speed up the analysis of the various traits. For that, we developed artificial intelligence methods such as deep learning for image analysis to increase data quality and speed up the image analysis. Manual annotation of many images provides the ground truth which is needed to test and validate our algorithm, which led to very fast analysis of images.
That's all, folks.
Okay, so that was the presentation by Christophe. And now Renaud will present results. So in this platform, the red and durum wheat panel of 250 lines has been analyzed several times. Yeah. Renaud, the floor is yours.
Okay, thank you. Thank you, Christophe. Can you see my screen?
Yes.
Okay, so hello everybody, I'm Renaud Rinçant and I will present you some results that we obtained by analyzing some 4 PMI experiments. And this data come from different projects, French projects, Archirac and breed-wheat. So we worked on more than 700 breed-wheat varieties, representing French elites, registered varieties, and worldwide diversity. All these lines were phenotyped in multi-environment trials in France, and each environment has been densely characterized, and in particular with some environmental descriptors. Each variety was phenotyped on the 4 PMI platform for roots and aerial traits in optimal conditions, and each line was genotyped with a dense SNP array from the breed-wheat projects. First, looking at the registration dates of the lines, we could see that there was some historical trends for some traits. For instance, a decrease in the number of seminal root numbers between 1940s and today, together with an increase in the number of tillers, a decrease of above and below ground biomass, and an increase of rooting depths. So these are slight tendencies, but they are significant, and they are probably due to breeding. So when looking at correlation between platform traits and agronomic traits measured in the fields, we observed a small correlation, most of the time not significant, and highly variable from one environment to another. When we combine all root traits together, we could explain around 10% of the variability for grain yield after correction for flowering time. We used the platform trait to cluster the genotypes into 4 clusters, and we could see that this clustering could explain parts of the genetic variability for grain yield, even after correcting for flowering time. Then we made some association mapping on the platform traits and also on the agronomic traits in the fields. And we identified some colocalizations and the root traits, which had an effect on agronomic traits, had a specific effect depending on the environmental conditions. For some of these QTLs, we could explain, or we could find some hypotheses using environmental covariates. You have here two examples. One QTL of rooting depths from the trait measured on the platform, which affected grain yield. And this effect depending on the level of hydric stress. The higher the depths, the higher the grain yield in hydric stress conditions. And so the effect was positive in some environment, in hydric stress environment, and negative in optimal environments. You have also an example of QTL affecting root biomass, which had an effect on grain yield, and this effect was dependent on the level of radiation of the environment. So to conclude, we could show that the recent breeding has indirectly slightly influenced root traits. We could find relationships between platform and field at the trait and at the QTL level. The effect of the platform QTL on agronomic traits were environment specific, and we could explain in some cases, or we could find some relationships with environmental covariates. This result will be hopefully soon published in Frontiers. Our perspectives are to work with more sophisticated traits that we could obtain using some image analyzing software or deep learning. Of course, using modeling to better model and understand the relationship between platform and fields, and considering the polygenicity of the platform traits to use some genomic selection. We have obtained some first preliminary results in SolACE, which are quite promising and which we'll present at the final meeting. Thank you for your attention.
Thank you very much, Renaud. So you start to see one of the messages we're probably going to bring from this project is the environmental specificity of QTL effect. So dissecting phenotype and the genotype is required, but also needs to take into account this. Well, that will be the point for discussions. So I'm now moving, and I will share the screen. So now coming to the second example of what was done in SolACE with durum wheat in this case, with dissection of root phenotype into phenes that are closer to root model parameters, as will be shown, and that can be used as it has been used as ingredient for genomic selection. So in a small video to illustrate the root fur platform that was used. So it's a small platform in Louvain-la-Neuve where we can accommodate 1,000 plants for approximately 3 weeks. So the environment in the greenhouse is mapped with a resolution quite high in terms of light, and we have a dedicated scanning system that will take pictures of those root growing in 3 dimensions. So these are high resolution pictures. And the roots, as you see, are growing in aeroponics, giving a very specific type of environment. So the platform is now in action. Every plant will be imaged 2 hours, every 2 hours. This will lead to a large number of images that has to be analyzed, and we designed a strategy based on tip detection. And you see each color point here is a tip that was detected, and they are overlaid on the final image. And by analyzing the tips as populations, we are able to estimate parameters describing growth, branching, angles, of the 250 lines. The point is that once we have those traits and those simulation, then we feed into the modeling part of the program, where we are able to simulate water uptake as a function of depth and type of soil, for example. So that was the short presentation of route FAIR, and I will now leave the hands to Gabriela. And Gabriela first and Nicolas just after, to present analysis of data coming from this platform.
Yes, good morning to everyone. Let me share my screen. Can you see it?
Yep.
Okay. So good morning to everybody. Today I'm going to show you our results obtained from the genomic association analysis done on the durum-wheat collection that was phenotyped in the root PhenoAir platform. So this is a table reporting the list of all the variables that were measured in the root PhenoAir platform. As you can see, I underlined 6 of them, which are root dry weight, shoot dry weight, emergence of the primary root, median diameter for embryonic root per plant, primary elongation rate, and seminal root angle, because these 6 variables gave us good results in the GWAS analysis. Indeed, we were able to identify the chromosome QTL regions, and also to identify the percentage of phenotyping variability, which is explained by the R-square parameter. In the next slide, I reported the Manhattan plots I've obtained from these 6 variables, in which we tried to make some QTL colocalization between the 6 variables that gave us good results. In particular, this was possible for root dry weight, which seems to colocalize with the shoot dry weight on the 6 and 3b chromosomes. The same was able for emergence of the primary root with the primary elongation rate. Both these variables seems to have some SNPs in the 3b chromosome region. Then we tried to make some comparisons between different platforms. Indeed, the durum-wheat collection was phenotyped in 3 high throughput root platforms: root PhenoAir, the 4PMI, and the pouches experiment. So for us, it was interesting to see if there were some QTL colocalization between same variables measured with different platforms. We were not so lucky with this because we didn't get any exciting results except for the root dry weight that was measured with both the root PhenoAir platform and the 4PMI platform, and it gave us the result of a colocalization on the 3b chromosome. But the same was not possible for similar variables measured with different platforms, like, for example, seminal root angle and primary root angle. And an explanation of this could be the fact that these variables were performed and measured with different protocols and methods. But we still need to go into more details with this. And so for now, we will go on with the results, the analysis of the results we've obtained with the root PhenoAir analysis in the GWAS Analysis Association. So thank you for your attention, and that's all from me.
Thank you very much, Gabriela. So we leave now the hand to the next step of this analysis on durum width by Nicola Piccioni. So Gabriela, you probably need to.
Yes, you need to stop this. Okay.
Great, thanks.
Okay, can you see the screen?
Yes.
Okay, so what I present is I don't know if you can see the slide.
Yes.
What I'm presenting is the first results of Paolo Vitale, our PhD student that is trying to insert the root traits in a multi-trait genomic selection approach to support genomic prediction for grain yield in durum width. So this is the aim. The aim was to explore the potential to increase prediction accuracy through modeling of root-related traits in multi-trait genomic prediction framework. And for this, we used the SolACE data. The SolACE data from the two field experiments that were done in Italy at CREA of Foggia in 2017 and 2018 in this season, in which we had no stressed condition, well watered and nitrogen, and the stressed condition in southern Italy in rain-fed and minus nitrogen. So in this with these data sets, and also we used trace from 4PMI and from pouches to evaluate the possible insertion in the models of genomic selection. So when Paolo went to assess the accuracy of genomic selection for grain yield in stress condition, he found an accuracy of a little bit higher than 0.55 about, and using different models of applying different models of genomic selection. And but when we try to use as covariate 5 different best root-related traits, and in the multi-trait genomic selection using different models, we had not at the moment not an increase in not a significant increase in the accuracy of prediction for grain yield in the stress conditions. These are the best correlated root traits correlated with the grain yield in the stress condition. The accuracy just improves if you use as a covariate the grain yield in non-stress conditions with all the models that we have used, and with a slight difference while using in the model with respect to the other 3. So as a conclusion of this experiment that we are running, so this was the way to improve the accuracy for grain yield in stress conditions, more than with the root trace used in the phenotype in the platform, in the controlled environment platforms. And what we are doing now is just to use other ways for inclusion of root traits in the genomic selection models, like using a machine learning random forest algorithm, and also deep learning densely connected networks, to see if with this machine learning algorithms we could improve the result for the inclusion of the root traits in the multi-trait genomic selection. And that's all for the moment, and thank you.
Thank you very much, Nicola. I'm sure there will be further question coming in the discussion when we try to start predicting from including route information. So we will now move to more whole plant story with a presentation by Boris Parent of an experiment that was carried out in the PhenoArch with, in this case, a subset of this 200 panel. Boris.
Hi, everyone. I'm trying to share my screen.
Yes.
Is it okay from your side?
We are in presenter mode.
Okay, it's because.
So probably if you switch the screens.
Yeah. And now?
Still.
We did that 1 hour ago. It was.
It was working.
Yeah. I never understand how it works with several screens.
So maybe if you first, so if you unshare, then start launch the presentation.
Yeah, it's what I will do.
Then share again. Then you can select.
Maybe you can go with un paramètre d'affichage et aller dupliquer l'écran.
I will put my screen here. And this one is a good one.
This one is a good one.
Okay. So yeah, I hope I will not be too much out from the field, because I will talk mostly on above ground traits. And so the main question was which genetic trait we measure in the platform and how it is linked with what is observed in the field. Because in the field, what we have for measuring or for analyzing the response to drought is yield as a routine. So the response of yield to any constraints in the field. And in the imaging platform we have in Montpellier, what we can do really easily as a routine is to measure shoot expansion and then to have the response of shoot expansion to drought. So we wanted to link the traits, and to answer the first question is if there is at least an interest of linking them, which information the platform can give, and to have linked with the genetics, and can we use the trait we observe in the platform to predict or to infer the effects in the field with crop modeling, for example. So the strategy we had was first to analyze what was the response of yield to drought, which component was affected. So yield, the yield component, grain size and grain number, and for grain number, the ear number and the grain per ear. This, and to link that with what we observe in the platform. And shoot expansion is the result of three underlying components, which are the leaf development, individual leaf development, individual leaf expansion, and number of tiller. So it's what we try to answer. So the first question, the link between the response of the yield component and that of yield. So that's the result of the SolACE in the field. I just want to show you one row. Each row it's one site with for each component the value in the high inputs in the x-axis, and the y-axis is low inputs. So for example here we have a strong effect of the low input treatment. We are far from the first bisextractrice. So there's a big effect of the low input treatment. And this effect is due to the number of grain and the weight of one grain. And so here you can see that it's mostly due to the grain number. And this grain number is mostly linked to the ear number and a bit less to the grain number per spike. So that was the first answer. The sensitivity that of ear number in the field explains most of the variability that we can observe for yield. The second thing that we observed was, so in the platform, the link between the response of shoot expansion, the whole plant shoot expansion, and its component. So in the platform experiment, we had 90 genotype, the half of durum and the half of bread wheat. And because of the result of the field, we selected genotype which had contrasted sensitivity for yield. So with sensitive genotype, the red one and the blue one are less sensitive genotype, and but that for different reasons. So we take genotype on the right and on the left to have different sensitivity of grain per ear and different sensitivity of ear number. And so what we compare is for shoot expansion and its three related components, so leaf development, individual leaf elongation, and tethering. And for each of this process, we compared integrated value matrix, which has in general the maximum value, the rate, so the progression over time, the rate and the sensitivity of this rate to drought. So I go rapidly for that, but it's what we've done for each trait. So that's for one genotype, you have each process and the value, the absolute value, and then the calculated rate, and then the calculated sensitivity to soil water potential. And it's what we've done for the 90 genotype. And what we observe is first, when we speak about absolute values, and under well-watered conditions, for example, you can see that the diversity that we observed for shoot expansion rate here is really higher than that of leaf development rate and leaf expansion rate. And the higher variability, the diversity, was for tethering. So the diversity that we observed for shoot expansion rate was mostly explained by that of tethering. And we try also to understand which component could explain the sensitivity of shoot expansion to soil water potential. So we linked this shoot expansion to the sensitivity of the three components here. And if we look at what we observe for, so that's for bread wheat, the blue bars are the sensitivity of tethering, the red, the sensitivity of leaf appearance rate, and the green, the sensitivity of leaf expansion rate. We can see that the sensitivity of the whole plant shoot expansion is mostly explained by that of tethering, and it's in probably all genotypes, and it was also observed in durum. So of course we can observe correlation between the sensitivity of shoot expansion rate here with that of tethering, with the best correlation was between tethering, the sensitivity of tethering and the sensitivity of the whole plant shoot expansion. And then we try to link this result in platform and in the field. And so the first comparison was between shoot expansion measured in the platform and in the field with drone or with Phenomobile, and also with the yield component in the field. So that's the preliminary result. So this is a PCA with data variable observed in the platform, the blue one, and in the field, the red one. First, what we can say is that, yeah, a platform is different from a field, and traits are different. But if we look into details, there's one correlation which appears quite clearly, is the correlation between everything related to growth in the platform. So shoot area, shoot expansion rate, maximum number of tiller, etc., and GAI, green area index in the field, and the rate, the progression of green area index in the field here, and its maximum value, etc. And it appeared also when we did a clustering of variable, of all this variable, and what appears is that we have the rate, the progression of green area index in the fields is in the same cluster as shoot expansion rate and maximum shoot area in the platform here. So there are really preliminary results. We are currently working on the sensitivities and to integrate also the sensitivities in the field into this analysis. So the conclusion is that the sensitivity of here number was explaining most of the variability of yield in the field. The diversity of shoot expansion rate was mostly explained by that of tillering, and also the diversity of the sensitivity of shoot expansion was driven by that of tillering. There's really a strong link between tillering and shoot expansion. We had a strong link between shoot expansion measured in the platform and green area index in the fields, and poor correlation between this component in the platform and the yield component in the field. So we are still working on that. So thank you. And I just want to give a message. This work has been done mostly by a PhD student, Stéphane Levaux. He's currently finishing. So that's his email address in case you're looking for a new postdoc. Thank you.
Thank you very much, Boris. So yeah, it was a presentation about the shoot part, but tillering and number of nodal roots in wheat is certainly not completely independent. So we expect that what Boris has been talking about is probably closely related to underground stories as well. So this being said, I will now leave the floor to Pierre Martre, who is our last speaker and will present the work that Pierre, are you with us? Pierre was with us.
Yes, sorry. Hello.
Hello.
Hello, Xavier. I was lost with fighting to find the Zoom thing, the bar. Okay. May I share my screen? Yeah, I'm looking.
Yeah.
So I guess you're not seeing the right screen, right? Is that correct?
No, we are, yeah, presenter mode. Yes, that's fine.
It's better? Okay. So, okay, let's start with from the beginning. So hello everyone. Yes, so Xavier asked me to present you some of the work we've been doing in SolACE and mainly focusing on modeling strategy. So I'll try to do that in 5-10 minutes. So first thing is something, I mean, everyone working on in the field and measuring traits in the field, or even in control conditions actually, see the difficulties to upscale simple physiological or architectural traits to more canopy level traits, and where that scale usually at which we're really interested in terms of yield, protein content, or nitrogen use efficiency, water use efficiency. So the things that, because of and that's the interactions between the environment and management and the genotype that makes the connections between simple architectural or physiological traits and the more complex traits of interest difficult. So how could you try to find our way in this very complicated road between this simple and complex traits? So one option that has been proposed is to use process-based model that decompose how these simple traits that characterize the genotypes will affect these complex traits through a series of feedbacks and processes and regulations. So you have many important feedback that act at different timescale in the plant. I have just summarized a limited number here that are represented in a crop model. For example, of course, you will have a positive effect of the intensity of the solar radiation and photosynthesis in the field, at canopy level at least, and that will tend to increase the carbon reserves and in the short term to increase the growth of the plants. But if you have accumulation of carbon, that will have a negative feedback on the photosynthesis. On the other hand, the carbon reserves will have a positive feedback on the nitrogen assimilation and on the root growth. And the nitrogen is acting the other way around, is that when you have plenty of nitrogen in the soil, you will start, or you have reduced growth, you will start to build up nitrogen reserves, which will have a positive effect on photosynthesis, will stimulate photosynthesis, but then there will be a downregulation by the nitrogen reserve on the assimilation. So the plant is keeping a number of feedbacks like that, try to stay kind of balanced in its different components. And that's what we try to represent in the model, is this type of feedbacks and regulation that explain why. And this explains why it's not easy to go from these simple traits to more integrative traits, because of this cascade of different feedback that may have a different time scale and different spatial and temporal scales. So how can we represent the genetic variabilities in a model, in a simulation model? Well, a simulation model, crop model, or an architectural model is no more than a series of response curves or transfer allowing curves of more physical or mechanistic processes that relate one variable with another, and that can be either time or it could be a function of environment. For example, if you have a relationship between photosynthesis and light, we know that it has this type of curve, but it could be also a relation between a variable, let's say photosynthesis here, and another variable or here, or stomatal conductance and ABA, for example. So the way we can put genetic variability in here is through the parameters of this relationship, because different genotype may have different values for some of the parameters that define this this relationship. And what we try to do in the type of model we developed in process-based model is to have parameters that have biological meaning, and as much as possible we try to have parameters that we can measure directly. And so you have a number of so-called genotypic parameters that will be a characteristic, a fingerprint of a genotype. And this series of equations with this parameterization will allow you to predict the response of the system to perturbation from the environment, or from the genotype, or from the management, which is part of the environment. So here is a representation of the crop growth model, where you have different parts. It's very much simplified here, but you have different parts with mechanism, the canopy levels, with many interactions with the grains, also with photosynthesis and allocation of carbon, with the energy balance of the canopy and transpiration, but also of course with the soil. What you could see here is that the roots, there's no compartment for the roots. Actually, it's hidden in the soil, because the description is it's not very detailed description in most crop models. This is the example of a model called CREA quality, but it's the same for most crop model where the root system is not really representing. What we try to represent is kind of emergent properties of the root model. In the case of this model, and that's the same for AppSim, so we work pretty much the same way for the root system. So we have two main parameters. One is the rate of vertical root extension, so how fast the root front goes down the soil profile, and the other one is a root system occupancy factor, which represents the relative rate of root how the relative rate of root water extraction or nitrogen changes reduces with soil depth. So it's a mimicking changes in both in the hydraulic conductivity of the soil, but also on the root architecture and distribution, vertical distribution of the root system. And with this, with this simple parameter, we can already explore the impact of this macroscopic, let's say, traits. Here is an example of simulation where we analyze the sensitivity of the model if we change to the change in root vertical distribution and root front velocity in two very different environments. One in the UK in Rothamsted, and the other one in Spain in Seville. What you can see here is that depending on the level of stress here represented by the drought stress environments, so the level of drought increases when you go from the left to the right, and you see here that in both of these environments, if you have a deeper soil under drought stress, but also under non-drought stress here, you will have a positive impact on the yield. Not so much in the UK, although in more extreme droughts you may have some effect, but it's still very limited. And if you change the root front velocity, so how fast the root will grow downward, you see that for actually in Sevilla, you may have quite substantial increase in yield if you actually reduce the rate of root growth, which is quite counterintuitive, but actually because it's a strategy to store water, to save water for later in the growth season. And when you analyze this simulation, you clearly see that the genotype that will grow too fast in the root system, then they will use the water too fast as well. So I will not go much in detail here, but if you do a detailed analysis, you will see that depending on also on the environment and the years and so on, the effect of this trace may be positive or negative. You see that even under very high drought, severe drought, for some years, this effect of this trace here, if you have a more slow growth, you have a positive impact. So, but the things that you have no explicit representation of a root system architecture and responses to local water or nitrogen in this model. And so what we are doing in SolACE is that we have coupled this crop growth model, or in fact, we replaced the very simple representation of the root system in the crop growth model by the serious quality. In the serious quality by the 3D architectural model called ArchiSimple. And the main properties of this model is that the rooted diameter determines its potential elongation, and the diameter of mother roots determine that of local roots, and the actual root elongation rate is determined by the carbon satisfaction coefficients, and that the root system carbon demand is proportional to its biomass. And so the main processes simulated by ArchiSimple are the rate of root emission, the root elongation, and branching and root decay. And so most of these traits here, all of these traits and other traits here, like the root tip diameter, are parameters that we can actually phenotype in phenotyping platform. Also, we can measure the root elongation rate. So a number of parameters in this model, we can measure them at a relatively high throughput in the platform. So what we did also is that the representation of soil in crop model is rather simple in the way it's usually using a 1D description of the soil, and the physical processes are not directly represented. We rather use a tipping bucket approach to represent the transfer of the water through the soil profile. And so we replace the simple soil model, a tipping bucket model in series quality by a 3D general purpose reactive transport model called the Min3P. And so what we're doing here now is that we are using some of the data that have been presented earlier today from the root phenotyping platform to parameterize the ArchiSimple model. And the idea now is to do the simulation with the different traits in different set of environments. So we can try first to reproduce and to predict difference between genotypes in experiment carried out in SolACE, like in the ReadyMax platform in Copenhagen, but also we can explore the response of different combinations of traits at the European scale or different scales. That's the ongoing work we're doing at the moment, is to implement the root response plasticity to look on nitrogen and water supplies, and at the same time I'm trying to parameterize the couple model for the core panel of the breadwheat cultivar phenotyping work package 2, and then to analyze by simulation the value of the above and below ground traits in a wide range of environments, starting with the SolACE experiments. Thank you very much.